Hey there.
Hello, hello.
How
Hi.
how are you doing?
Sure, I was doing good, and then I checked my stocks, so
Ha 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 ha.
there's a new date. New
Yeah,
date tomorrow.
me too, actually.
I don't know what happened today, but
It's been like this for like the entire 2 months. Like
yeah, it goes up and then down. I'm like, I don't know. It says
most
volatile.
mostly down. It's been
Yeah.
like, what are you in? Like, what are some of the things you're in?
So I bought in like 2 weeks ago when the market, or like 2, 3 weeks ago when the market was down, but like MicroStrategy, like the Nvidias,
Uh,
Microsoft, a lot of the tech stocks like CrowdStrike, AMD.
yeah.
Amazon, mostly the tech stocks. So they were
Uh-huh.
like, remember when the market dropped like 20%, like back in March 10th, 11th? I had some money, so I just put it all in. I'm like, screw
Oh,
it. That's probably gonna go up. But
yeah.
then it just keeps going up and down. I'm like, whatever, I'm gonna stop looking at it.
Yeah, no, if you got in March 10th, then
Yeah,
that's great. Like, because
yeah,
if you had it prior to March 10th, that's when you like lost a lot. So like,
yeah. Because I was seeing the chart was like it was super high,
mhm.
like when Trump was in town, and then
Yep.
just boom, everything crashed. So
Yeah,
like,
especially
might as well.
with with MicroStrategy too, because we were waiting on Trump, you know, saying something positive about like the strategic Bitcoin reserve, but
Yeah,
ended up just being already confiscated Bitcoin, meaning that we're not spending money to buy Bitcoin. So that's like when I just like dropped most of that stuff. But yeah, you got in at a
yeah,
very good time. But yeah, it's you still got in at such a great time that
yeah.
like that you're pretty much good. But yeah, I put in.
I was trying to do like I was trying to figure out options and stuff, but like
Oh
I
yeah,
don't know. It's like that like gambling stuff is confusing to me. So
oh yeah, that's like
I don't know.
what I have here too. Like right now, it's gonna look. Let's see if you could see it.
Oh, okay.
So these are like the on the left, these are options, and pretty red today, but that's just what happens. But for instance, I have like so like Geo. So these are kind of like the stocks that went up when Trump got elected. So Geo is in charge of making private prisons, and with Trump, there's a lot more like people getting sent to prison and stuff like that. So I have some Geo. I have some other stuff, Archer Aviation, some gold and silver are really
Oh yeah.
good. Yeah, my and I have like another over
So
here.
like how does it work? Do you like buy it out for like I think it's gonna go up in like a month or so or
Yeah, yeah. So for instance, right, like I just put in this Nvidia order, and with the option expiration is August 15th.
Oh, okay.
Yeah, so it has that much time to get to where,
That price mark
yeah, yeah.
before it like goes useless.
Yep, yep.
Or
And here on my right, I have like my actual portfolio that I just turned into a spreadsheet to see that there's like so these are some of the stuff that like I'm in, and you can see like uh
yeah, Palantir, I was looking at that one too.
yeah, so I remember when
Yeah.
Palantir went up like to like 140 something like that, and then it like shot back down. So
Hmm.
you want to be very careful with things that are like
Like
oh,
that volatile. Yeah.
that are oversold. Uh,
Yeah.
so like for instance, like Seeking Alpha is a pretty good website. They have lots and lots of analysis and
Mm.
stuff like that. But yeah, on the left are my options. Today I just put in, I mean, in my actual portfolio, like I have like for where's
Yeah,
Nvidia.
Broadcom. The best one for me was the CrowdStrike that I got.
Oh, I made a lot from
Yeah.
shorting it um
Oh yeah.
earlier, uh but yeah, Nvidia I bought at like 114.70. It's like below that now, but I opened up a new option for it here and for AVGO as well. So like this like tells me I also like put in like the date and price of the recent dips. So I get
Um,
kind of track of like, if the dip if the price is below the dip, this part's going to be green. So,
hmm.
but yeah, you could see like the most like
So it's
to get all green here. These are just like from mostly like gold and silver and stuff.
yeah, so you're not like that risky doing options because you're doing it so far out that it's like I don't know. Because I just look at Wall Street bets and I see these guys losing their whole portfolio in like one day. So
Well, that
I'm
that
like,
certainly happened because like it's ironic because like a couple of maybe like 2 months ago, I was saying like, okay, options are impossible to lose because you could just have it uh expire from a really, really long time from now. But that's assuming that a stock is going to go back up. But I was in like, for instance, Lunar,
hmm,
intuitive machine,
yeah,
right? And like in the morning, like I listened to like Bloomberg and stuff, like they went to land their moon, like a ship, like a spaceship on the moon, and when it landed, it tipped over, and
hmm. Oh, and, oh yeah, yeah, I heard about that, and then it just sank that stock.
yeah, it dropped 50%, and that wasn't coming back. And then with quantum stocks, when Nvidia guy said, we're like 20 years away or 30 years away,
Yeah,
all of the quantum stocks dropped like 50% also. So I definitely got pretty cooked. But
yeah.
it
The
doesn't
thing is, they all go down. Like all those tech together, they all tank together. But then, like with AMD and Nvidia, I feel like one of them goes up and the other one kind of goes down. So I don't know. It's like competition or something.
yep, that's why it's hedging, right?
Yeah,
So with yeah, yeah. So
yeah. I'm
crazy.
still getting into it. I'm just blindly following stuff. So,
Yeah, yeah.
but
I could even share like my portfolio like with you, which shows like what's how much of each asset and stuff. Because I can
okay.
share that with my broker. But
Yeah,
yeah, yeah.
I'm done.
All
I
right.
don't know much, so we'll see.
Mhm.
Let's get into
Yep.
the more exciting stuff.
Yeah.
So I think this is a question on the full length. I'm not sure if I don't think we really need the passage for this one. It's talking about what's like least likely to occur in a muscle fiber that's metabolizing anaerobically. So I know like glycolysis, or no, like anaerobic is like lactic acid, like fermentation that happens anaerobically. But like stuff like glycolysis, you also don't need oxygen to for it to work. Is that kind of
Uh,
what this is talking about? Or
yeah. So anaerobically means, without oxygen, right? So you're gonna have um like glycolysis, and after glycolysis, when you have your glucose turn into pyruvate, the pyruvate, if there's oxygen present, the pyruvate will go into the mitochondria and undergo,
hmm,
the pyruvate dehydrogenase complex, the TCA cycle, the electron transport chain, right?
hmm,
The proton gradient, the ATP synthase. So,
yeah.
but if there's no oxygen, then the pyruvate gets turned into lactate, which could help replenish the amounts of NAD plus needed for glycolysis, and the lactate can travel to the liver via the Cori cycle to uh, undergo gluconeogenesis to create more glucose and bring it back to the muscle, to be used for glycolysis, right? So, which of these choices talk about uh, something that happens with oxygen?
With oxygen, said the electron transport chain.
Yep.
That's one. And then
Yep, that's the only one, really.
I think what I had trouble with is like figuring out what these were talking about. Acidification of the cytoplasm. Is
Uh-huh.
that like just I know glycolysis occurs in the cytoplasm.
Yep.
So maybe it's least likely to occur there. And then depletion of stores of glycogen, so glycogenolysis.
Well, you said that what happened that you so the lactic acid fermentation, the lactic acid, uh, the pyruvate turned into lactic acid in the cytosol, right?
Oh, okay, okay. Yeah, yeah, yeah.
So that would
So
cause.
instead of
Mhm. And then since glycogen is, polymerized glucose, and you're trying to get more glucose to be made, you
mhm.
could be breaking down glycogen also.
Okay. I got this one right. I just didn't really know C and D, like kind of the explanation for it.
I see.
Yeah. I just wanted to make sure I understood the concept. Okay. Yeah, this question uh, decides to load. Which of the following animal pairs best illustrates the outcome of convergent evolution? Well, from what I know, convergent evolution is like two like unlikely species coming together to have similar stuff. I don't know.
Yeah.
That's how it is. But then it gives me like these like animals and stuff, and I'm like, how am I supposed to know like what's similar and difference between them?
So convergent evolution is when two separate species end up having a similar trait, right?
Yeah.
So like an example that I can give that's not here is a bat and a bird.
Okay.
They both have wings and can fly, but one is a bird, a species of, bird, and the other is like a mammal, right?
Okay. Yeah.
So here,
So
uh,
for A, the dolphin and the shark, they're both in like water and they're both mammals or something. I'm not good with stuff with stuff like this.
so let's think, right? So, uh, sharks have gills, right?
Yeah.
And dolphins, or maybe how about this? You are you more likely to see a dolphin like jump out of the water than a shark?
Yeah.
Okay. Now, if we think about that, why do you think that would be the case?
Uh,
If you were underwater, what would make you go back up to the surface?
to get some oxygen or something.
Yeah. So if you've ever noticed, dolphins and whales, their tails go up and down, whereas a fish or a shark, the tail is going side to side, right?
Yeah.
So now when you pair that with gills, you could see that a fish or a shark, because they have gills, by the way, get their oxygen from the water through their gills. So if your tail can only move side to side, you don't really get a way to quickly go up and down, which you wouldn't need to do if you already got oxygen from your gills, from the water passing your gills, right? But
Hmm.
for a dolphin or a whale, their tails go up and down. So you see them a lot of times moving up and down in the water quickly and jumping up for air because they don't have gills because they're mammals, which means that they give live birth, um, and more intelligent, etc. So when you look at these choices, right? So D is saying the same species, peppered moth.
Yeah.
One is light, one is dark, but they're still the same species. Polar bear, panda bear, they're not exactly the
Yep.
same species, but they're part of the same family. They're bears. Domestic
Okay.
sheep and the mountain goat as well, are similar species, right? With a similar, genetic lineage. But the dolphin and the shark are very, very are not just different species, they're different like I don't know if it's like I forget like if it's kingdom or class or fan.
Oh.
But yeah, yeah, because the shark, which by the way has lived from before the dinosaurs and still lives today without any evolution, like the shark has not changed in like 70 million years. Like it's like pretty much perfect the way it is. And yeah, a shark you could think of, like it stays in the water, it doesn't really jump out for oxygen, it has gills, it yeah, stuff like that. And then like the dolphin, is more like whatever, intelligent and is a mammal, gives live birth and can go up and down. But it would need to go up to the surface to get some oxygen, same way that we would, because it's a mammal just like we are. Uh, and also like warm-blooded, I guess. But yeah, so they both can swim. They both have, fins and stuff like that, but they're from a different genetic lineage.
So yeah, different taxonomic families.
Mhm.
Okay. Yeah, I was just so confused on that one. I was like, what does this have to do with glycolysis?
I mean, like, interestingly enough, sharks can even like detect electromagnetic radiation that's given off by, you know, your body when you're moving around because of your nervous system. But yeah.
Yeah. I think we can just go through this one, break it down.
Mhm.
So butyrate, a product of bacterial anaerobic fermentation, remember that, is a known regulator of gene expression. For example, exogenous butyrate is used in clinical studies to stimulate fetal hemoglobin expression in individuals with sickle cell anemia. Butyrate exerts its function by inhibiting histone deacetylases,
All right.
which counter.
Sorry. Uh, yeah. So this is starting to get into like, lots of, stuff thrown at us category. So we should write some proportionalities,
Yeah.
right? Because they're talking about like this causes this and this inhibits this, and that's really hard to understand. So, okay, so butyrate, when they say like that it's
And
that it's um okay, so we could write like whoops. Butyrate is proportional to like hemoglobin fetal expression by inhibiting HDACs. And see, like, and now this is why this gets confusing is they're saying that butyrate inhibits HDACs, which counter the effect of HAP. So if they're saying HDACs is inversely proportional to HAPs, butyrate, which is inversely proportional to HDACs, must be proportional to HAPs,
hats. Yeah.
which they're saying are histone acetyltransferases, which I guess, transfer acetyl groups to histone. And well, also, what can you tell me about histone acetylation?
It's when you make it make like euchromatin or like you expose the lysine residues to make it easier to bind for transcription.
Yep, yep, yep. So
Yeah.
yeah, so uh, like the beads on a string with the histones, and acetyl groups when you add them push those beads like the histones further away, which means more DNA between them is exposed to transcription factors, which can then transcribe them into RNA and translate them into protein. So, uh,
Histone deacetylase will condense them and prevent the transcription.
yeah, I think so. Yeah.
Yeah. Okay.
So chromatin, and then they say that these modify basic residues on target proteins. Okay.
Okay. All right. Butyrate is structurally related to beta hydroxybutyrate, the major source of energy for mammals during prolonged exercise or starvation. Oh shit. To determine whether uh, BOHB may also have HDAC inhibitor activity, researchers treated cells with different concentrations of this. After 8 hours of the exposure, histone acetylation levels were measured by immunoblot with antibodies specific to the H3 histone acetylated at lysine 9 or lysine 14. The antibody only binds to acetylated tubulin. The results are shown in figure 1. So it's telling us the acetylation level. This one for K9 is has a higher acetylation level. So,
So,
oh yeah.
so, so we know that butyrate um, can cause, the acetylation. And now we're trying to find out if beta hydroxybutyrate also inhibits HDAC. And, they're saying that it's for mammals, so I guess that's how it's different from the regular butyrate. But yeah, we're trying to find out, if this has HDAC inhibitor activity. And so so I guess they treated cells with different concentrations of this, and after 8 hours, they measured acetylation levels by using these antibodies, um, which only bind to acetylated tubulin. So yeah, okay, so then our x-axis is the concentration of beta hydroxybutyrate, y-axis is the um, amount of protein acetylation level. What do you think they mean by fold?
Uh, like the level increase,
Yep. So like at 10,
like
it'd be tenfold, meaning 10 times as much.
oh, okay.
Mhm. Like I remember my friend, there's a band called Avenged Sevenfold. I don't know if you've ever heard of them,
No.
but Avenged Sevenfold, it's they sometimes write their band name as an acronym that's an a 7, and an X. And my friend was asking like, why is there an X? And the X means fold. So if I had four times difference, that's fourfold.
Yeah. Okay.
Okay, so here we see that for wait, the antibody for this only binds to acetylated tubulin. Okay. Acetylated tubulin, acetylated K14, I guess, and then acetylated K9. So maybe I would do like in terms of acetylation, like K9, K14, K40, something like that.
Okay. All right. Researchers also identified a role for beta hydroxybutyrate concentration and protection against oxidative stress, which can result in carbonyl derivatives that are formed by direct metal catalyzed oxidative attack on amino acid chains of proline, arginine, lysine, and threonine. Mice were implanted with an osmotic pump that delivered phosphate buffered saline. These mice were injected with paraquat, which increases reactive oxygen species as or a vehicle controlling injection. After 2 hours, kidney samples were isolated and protein carbonylation was assayed by immunoblot. These results showed that protein carbonylation was suppressed in mice receiving beta hydroxybutyrate. Okay,
Let's
so
talk about what this is saying.
uh, we're saying that the mice were injected with this paraquat, which
So,
increased.
so
Uh,
start at the top when you do like
yeah. So it says they identified a role for the concentration and protection against this oxidative stress by, and then it results in carbonyl derivatives that were formed by oxidative attacks on these amino acid chains.
huh? So when they say oxidative stress, what do they mean?
Uh, Something with oxygen, like protection against oxidative stress.
You know what free radicals are?
Not really. I don't know how to explain it.
What about antioxidants? Like maybe like in some food, like it'll be it'll like show like, oh, it's full of antioxidants, so it's healthy. Like, have you ever seen
Yeah.
that? So okay, so this is important. So a free radical is something with an unpaired electron. So
Okay.
electrons like to be in pairs. If they're not in pairs, they freak out and they get super reactive and bad. So a free radical would be something that has a an unpaired electron and they're super reactive and damaging. And another way of mentioning or another way of saying uh free radical is reactive oxygen species, ROS.
Mhm.
See that? That means the same thing as free radicals. And antioxidants can give electrons to free radicals, therefore making the free radicals, kind of neutralized. So that's what they mean by oxidative stress and reactive oxygen species, because they say that um, you know, uh, the beta hydroxy butyrate, so I'll write like beta OHB inversely proportional to ROS. Wait, let me make sure. So, yep, protective role against oxidative stress, which is ROS. And they use something called paraquat to produce, I guess, reactive oxygen species for the experiment. And then they looked at kidney samples and the carbonylation. Um, and oh, and then they said that it was so wait, carbonylation. Not sure what that does. Maybe it, but okay, we can see that carbonylation was suppressed in the mice receiving beta OHB. So I'll put this 1 over protein carbonylation. So, yep. So this is what I would write out. I certainly wouldn't like not write it out because it's confusing like to keep track of like all of these different acronyms. So, but we can see that butyrate can inhibit HDACs, which means that it will like stimulate hats. And if you have more hats, you have more acetylation. If you have more acetylation, you have more expression. So we have a higher expression level of fetal hemoglobin once we have butyrate. And then they wanted to test this for beta hydroxybutyrate, and they find, different levels of acetylation, depending on which like lysine residue they acetylated. And then we also know from the last paragraph that beta hydroxybutyrate also protects against ROS. And yep, that's pretty much that. Mhm.
So for this question, if HDAC inhibition by beta hydroxybutyrate is a physiological response in living animals, the information in the passage indicates it is likely to occur when. So HDAC inhibition. So it says here it's the major source of energy for mammals during prolonged exercise or starvation.
Okay.
So if we were to, yeah, I'm confused. I'm not really sure what this question's asking, to be honest.
So, uh, sorry, I was responding to an email. So, okay. If HDAC inhibition by beta hydroxybutyrate is a physiological response in living animals. The information in the passage indicates it is likely to occur when. Okay. What did they say beta hydroxybutyrate was?
The major source of energy for mammals during prolonged exercise or starvation.
Okay.
So during, so like after, so it's like after starvation, your glycogen stores get depleted and it goes to like gluconeogenesis and ketone bodies and stuff like that.
So, when you're exercising, right, you first you'll first use the glucose in your blood, and then you'll use the glucose in your muscles through glycolysis,
Hmm.
and then you'll use lactic acid and the Cori cycle to produce more gluconeogenesis.
Okay.
And all of that stuff happens, but when you run out of that, meaning like carbohydrates and stuff and sugars, what will you start to break down?
Like the glycogen?
So after that, after you've already broken down the glycogen reserves in your muscles and liver.
5, 2, like beta oxidation and stuff like that.
Yeah. Right. Like your body will first break down the sugars because sugars don't serve any structural, purpose for the most part other than energy. Um, and but once you use all of that up, then you're gonna start breaking down like fats, and that's why if it's after sustained fatty acid oxidation, that's what would create beta hydroxybutyrate, which I believe is also a ketone body. Uh,
Okay.
but yeah, that's why it would happen then.
Could you write down the correlation that you did with the exercise? Like first it starts off in the muscle, like glucose in your blood, and then it goes to your glucose. You're using glucose in your muscles, which is glycolysis.
Yeah, let me get something with that. I'm looking for something in the Jack Weston like cheat sheet or quick sheet because I remember they had a really good kind of graph of. I'm just Googling that. Okay, got it. All right, I'm putting it in the chat.
Okay.
Yep.
Phase one, well fed.
Yeah, there's nothing listed for origin of blood glucose here because this is when you have blood glucose.
So your glycogen stores quickly depleted here. And then
Yeah, and you see.
body starts using gluconeogenesis.
Okay.
And then Stage 4 is when you use your ketone bodies, your fatty acid oxidation. And
Mhm.
so glycolysis happens like here,
Yeah, so think about it like this. So it's like, your oh wait, sorry, you said what about glycolysis? So
like when does it occur?
um so when you have the Roman numeral 1, phase 1, that's when you have, you know, blood glucose, and that can be broken down in like, muscle cells and stuff through glycolysis. And that and like it gets finished or used up 4 hours into it, and around that time in phase 2 you start to see um glycogen glycogenolysis, which means that this glycogen starts to break down into the individual glucose monomers. And uh, you also see that in addition to like the glycogen, because the glycogen seems like it's going down, right?
Yeah.
And that's because the y-axis is the amount of glucose. So if glycogen is going down, you can say that that's using up the glucose from breaking down that glycogen, and you're also having, gluconeogenesis happen, and that happens, around the same time. Um, but yeah, the glycogen reserves get used up by like 2 days into it, and the gluconeogenesis continues throughout the sol.
Okay. And then
Yeah.
during prolonged starvation is when your gluconeogenesis reserves go down and it's more ketone bodies, beta oxidation, fatty acid oxidation, stuff like that.
Yeah, yeah. I thought this was gonna list that, but
I kind of get the idea.
okay. Yeah, because I could have sworn that I saw this before with like the other breakdown uh products.
So like it's not increased gluconeogenesis because it's talking specifically prolonged starvation. While this is more, well, it's actually going to be decreased gluconeogenesis because according to that chart, it goes down. So, and there's going to be more fatty acid oxidation. Pentose phosphate shine is not really talk. It's not really in this.
Uh
And then,
yeah, pentose phosphate pathway makes nucleotides. So it's an anabolic process rather than
and
catabolic.
I'm breaking down. Yeah.
Yeah. But
Okay. Okay.
threw in another image in the chat that just kind of shows
Good.
I mean, actually the thing that I sent is kind of like wrong. Actually, never mind, because this is like power output and burning. So yeah, never mind about this actually. But
And then, what about the Cori cycle? That's not really. It's like happening. I feel like when your muscles are depleted. Isn't that similar to gluconeogenesis, kind of? Don't they? Isn't it kind of like a circle?
yep. So yeah, so when you finish using up your glucose and you turn it into lactate, the lactate can go to the liver and turn into glucose again through gluconeogenesis, and then that can come back into the blood.
Okay, that's good. We have time for one more question, or
Yep. Well, yeah, we started late, but we should have like 15 minutes.
I just don't know if you have somebody else or something.
Oh yeah.
Okay, this one we can just kind of go through.
Hmm.
A 2-year-old boy presented with short stature, weight loss, lactose intolerance, excessive flatulence, foul smelling, and fatty stools, and other intestinal symptoms. Okay. A biopsy of the boy's small intestine and specific antibody tests led the attending physician to believe it was like that child had celiac disease. To confirm the diagnosis, the physician placed the boy on a strict gluten-free diet. So this one's just talking about giving him symptoms of this boy, and they took a biopsy of the small intestine to, and then it's celiac disease. So, found in wheat and certain other grains, glutens are a type of protein that contains proline-rich regions that the human body cannot digest. In people who have celiac disease, these indigestible polypeptides are taken up by the mucosal epithelium of the small intestines, where they are deaminated by the enzyme TG2. Okay. Deaminated polypeptides often trigger an immunological inflammatory response that damages the lining of the small intestines. The intestinal villi atrophy, leading to a variety of symptoms, include those indicative of malnutrition.
What
So, for
that term?
the
Villi?
villi, that's like the thing and like that takes up the surface area or increases surface area.
Hmm?
And then atrophy means death,
Yep, for like weakening. Hmm.
weakening. So that kind of makes sense if you're not maximizing your surface area. That can lead to like malnutrition or you can absorb stuff
Yep.
as better.
Hmm.
And then within a days of starting on a gluten-free diet, the boy began to have an improvement in symptoms. After just a few months, a small intestinal biopsy showed normal morphology and the boy experienced a growth spurt. Okay. Next question I had, one consequence of advanced malnutrition is reduced amounts of plasma proteins in the blood. This condition will most likely cause the osmotic pressure of the blood to uh,
So what is osmotic pressure?
I just really know the formula for that, but I know it's higher. Uh, so is it like the same as oncotic pressure, or is that something else?
Uh, so uh, so the oncotic pressure is basically when you have. Okay, so let me draw something here. All right, this is a blood vessel, arteries here, capillaries here, veins here, and like the rest of this are cells, whatever. Now, at the so there's two types of pressure. There's hydrostatic pressure, which is how we typically conceptualize pressure, which is a pushing type of pressure. Like if you had like pressure inside of a container increase, it would cause pushing against the walls of the container. So hydrostatic pressure is going to be pushing um, water out of the blood vessels and into the cell, and we can call this filtration. And in the capillaries, it'll be kind of equal, but in at the veins, you're going to have an increase in osmotic pressure. So what that means is like, so let's say I have this like tube thing and I have like a semi-permeable membrane here that lets water travel rather than, let's say salts or solutes. Um, and let's say that we had like, high solute concentration on the left. You should see in a low solute concentration on the right, you're going to have water move from where to where.
From high to low, or
When you say that, so like high salt concentration, you're saying, or
I would want to go from here to here.
so if we have a high salt concentration, relatively, we have a lower water concentration. So the water on the right, which does not have any salts in it, will be sucked in past this semi-permeable membrane to the left side because the left side has more solutes, which means less solvent or less water. Maybe an easier example would be like, let's say that we have like H2, like some cell here with an H2O concentration of, let's just say 1, like an arbitrary number. And the outside has an H2O concentration, let's say like 50, and the cell membrane is permeable to water. What would happen?
The water, if permeable to water means water wants to go out.
So permeable to water just means water can pass through the cell membrane. So so in this box here is a cell with a low water concentration. Outside of the cell has a higher concentration of water. Now what can happen?
So wouldn't it want to go out? The the one on the inside,
Well, the one on the inside has a smaller, so like diffusion would go in which direction?
high to low.
So now if we have, so where do we
So
have
if we have a high out on the outside, it would want to go down
go inside?
in the inside
And you can have like lysis happen. Like,
when the cell bursts and stuff.
yep,
Okay. Is that
right?
like isotonic, hypotonic, or hypertonic, or like is that something else?
Yep. So this would be an example of hypertonic
Hypo-
where the solute concentration, well, hold on. I need to make sure that okay, so I there's a distinction I have to make between solute concentration and water concentration because typically we don't talk about water concentration, we talk about solute concentration. But the thing is, if you have high solute concentration, you will have a lower, relatively speaking, water concentration. So,
Okay.
so like here, so I could even write like H2O, let's say is 1, let's say sodium is 100. This is another cell. And outside H2O is like, let's just say 100. Outside sodium is 1. And the only thing that the cell membrane of the cell in the rectangle that I drew, the only thing, it's not permeable to the salt, but it is permeable to the water. What would,
So the water would want to go inside
yep.
here, but then the salt, since it's not permeable, nothing really happens, or what?
Uh, well, in real life, cells will be including like aquaporins and things like that that can help water pass in and out of the cell. So so yeah, in this situation, since you have a hypo, oh, and by the way, since we're that the term like tonic, like hypertonic, hypotonic, it's based on the solute concentration. So this outside area with a water concentration of 100 and a solute concentration of 1, this is actually called hypotonic.
Yeah, because then it like gets bigger, but then in hypertonic it like explodes or something.
Well, so
Or
hypotonic like this, water enters into the cell and can cause, like for bacteria and stuff, can cause that cell to burst because it's getting filled in, it's getting so much water sucked into it,
Mhm.
osmotic pressure, right? If it was the opposite, if it was a smaller concentration on the inside and a larger one on the outside, that would then be hypertonic, and that means the water will get sucked out of the cell and it can kill the cell that way.
So in hypertonic would it like shrink?
Hmm. Yeah,
Okay.
it'll like shrivel up.
Shrivel. Oh, okay, 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 okay.
Yep.
All right.
So now that's also what osmotic pressure is. So hydrostatic is like a pushing type of pressure, like particularly at the glomerulus, where like let's say this is the glomerulus, this is
Yeah.
Bowman's capsule. The hydrostatic pressure of the blood pushes against that Bowman's capsule to cause the filtering of that blood, right? So if we're going from our blood vessel to, a cell or organ or something, right? Uh, we're pushing fluid out from the blood to something else, right? And then in the veins, you're going to have a higher osmotic pressure. Oops. Because the uh because like the cells here are going to have a higher concentration of or rather sorry, the blood here will have a higher concentration of solutes, which means that it's the environment is going to be like um the environment is going to have a higher solute concentration, and therefore it'll suck the fluid out from the cells and back into the blood vessel.
So for the what's the A right here again?
Oh, artery?
Artery. So this is for hydrostatic pressure,
Yeah.
right?
Yeah.
Okay. So then that makes the cell bigger, or is this the cell or the membrane?
Oh, so I mean, so in the example that I wrote all the way here, this was like in the example of like if you're trying to like see the effect on a cell, like if it's gonna just like destroy it by bursting or shriveling it up. But
Yeah.
on the left, it's really more so just between like instead of it trying to destroy the cells, it's gonna like assist the cells. I'm gonna put a picture in the chat if you can open that. So we have here, I'll erase what I drew. So this arterial end has this, so it says fluid exits capillary since capillary hydrostatic pressure is greater than the blood colloidal osmotic pressure. So the like because you're gonna still have the combination of hydrostatic and osmotic pressure.
Yeah.
So it's just, which one supersedes. So on the arterial end, we have a higher hydrostatic pressure, which, comes from the fact that arteries, are high pressure. Um, so that gets pushed out of the blood vessel and it can go into like the cells, it can go into, the tubule system of the nephrons, all those things. Like if it's basically blood leaving a blood vessel and entering into a different cell or something like that, it's called filtration. And then in the middle here, we have like the capillary that's not like the arterial or venous end, and we essentially have an equal amount of hydrostatic pressure and osmotic pressure. So there's no filtration happening at the, middle of the capillary, but that is where the gas exchange happens.
Hmm.
Oxygen gets taken in, uh, and O2, sorry, CO2 gets taken out of the cell that is. But yeah, when we go to the venous end, we have a hydrostatic pressure that is less than the blood osmotic pressure. So it causes, and so I want you to think of osmotic pressure as a sucking type of pressure. And this sucking type of pressure, osmotic pressure, is greater than the hydrostatic pressure at the venous end of the capillary. And that's why you get reabsorption, which means you're taking fluids from the cells and sucking them up and putting them in the vein.
So that's just the result. It's just reabsorption. Like
Yep. But yeah,
so.
back. So yeah. And oncotic pressure is the same thing as osmotic pressure. It just specifically the osmotic pressure in the blood caused by proteins in the blood.
Okay, so how would you go about this question?
Okay.
It says consequence of advanced malnutrition is reduced amounts of plasma proteins in the blood. So like, should I go back to that picture?
So okay, so it's asking about so it's saying something about reduced amounts of plasma proteins in the blood. And so okay, I guess like I should have drawn this, but like the reason that the venous end has that oncotic or osmotic pressure sucking inward would be because there could be a larger amount of solute inside of the blood. So think about it like that, right? Like reduced amounts of these plasma proteins in the blood. What do you think?
So there's reduced amounts of proteins. And I'm kind of having a trouble trying to picture it.
Okay. So let's say that this dotted line that I put in the middle is what is a semi-permeable membrane that only water can travel through. Which direction would water want to travel?
To the right,
So which side has less H2O?
they're both equal, aren't they?
Are they? So like if you took
Oh, is it because Na plus follows H2O or something?
so the left side has within it water and a solute such as sodium.
Yeah.
On the right side, it's just water.
Okay.
So, so if you are so on the right side, it's 100% water, and on the left side, is it 100% water?
No, there's less water on the left side. Oh, okay, 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 okay. So it's 100% water on the right. On the left, it's not. Okay.
So,
We'd want to go to the left side.
yep, right? So, you could even think about like, do you ever like hear like someone saying like if you have a sore throat to gargle with salt water?
Yeah,
So that would so like if you have bacteria in your throat and stuff and you gargle with salt water, you're creating a hypertonic environment, meaning this thing on the left is the salt water, the thing on the right, let's say, is, the water inside of a bacteria. The salt water is gonna cause water to be sucked out from the bacteria and then like killing it that way. But yeah, the side that has more solute, because you mentioned that you remember like the formula like this.
yeah,
So if this is osmotic pressure, and for these types of questions, we're not going to change temperature or anything.
yeah,
Osmotic pressure should be proportional to concentration, molarity, right?
hmm
And this concentration is referring to the concentration of the solutes. And if you have a so solute concentration is going to be proportional to osmotic pressure,
yeah,
right? And so here in the question stem, they're saying that if we had less plasma proteins in the blood, and they're referring to plasma proteins as solutes here.
So if we have less solutes, we'll have less osmotic pressure.
Okay.
Then wouldn't that increase the fluid?
Um, in which direction?
Because we're bringing in like the water to the left side.
Uh-huh. So yeah, so if you have more plasma proteins in the blood, like okay, I could just do the same thing with, the cell here, the blood here, right? And the cell just has water, the blood has water, or actually, since they said reduced amounts, like we could say that the blood vessel just has water and the cell has, let's say, the solutes. Uh, that would be what this question stem is presenting to us, right? So which direction will the water travel here? And let's say that the blood vessel is also selectively permeable, letting water pass, but not sodium.
We'd want to go up to the cell
Okay.
because there's less water
Mm-hmm.
in there. So we'd want to go up to the cell.
Yep.
What's B again? You said the
Oh, like the blood vessel.
blood vessel.
Yep.
So would it be an increase?
Uh, in what? What direction?
So it's going up is the cell.
Okay.
The blood vessel would have a decrease,
Uh-huh.
but then the cell would increase.
Uh-huh. Yep.
Wait, is vessel same as tissue? I'm kind of
So it's always going to be relative to the blood vessel, right? And a blood vessel is going to be artery, vein, capillary. So
Mhm.
everything else that isn't that is the other thing. So whether
So
that's
okay.
cell, tissue, muscle, whatever, right? Just you want to see if it's because it's either going to be coming into the blood vessel or out of the blood vessel.
So tissue and all that is the other stuff.
Outside of the blood vessels, yep.
So it'd be an increase.
In,
The fluid of the tissues because it's going out of the blood vessel.
yep.
Okay. All right, take a picture of that. Let's see. Okay, 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 okay. Yeah, I never understood that concept till like now.
Yep.
Yeah. Okay, okay. All right. That's a good way to close it out.
Yep. So let's see. So for today, it'll just be 88. Uh, yep. And then I will be, yeah, I'm gonna see you on Saturday.
Saturday, yeah, I'll see you
All right then.
there. Okay.
All right, I will see you then, and good luck in your stocks and stuff.
Yeah, for sure. You too.